Principles of Inheritance and Variation, Session 7. Let us continue with the monohybrid cross.
In the next step, Mendel did selfing of the F1 hybrid, selfing or self-pollination. So, let us conduct the self-pollination. Capital T small t represents the F1 hybrid, and again a capital T small t represents another F1 hybrid in which they were selfed. So, let us represent it as selfing. Okay. So, again, what has to be formed? Gametes has to be formed. Are represented inside a circle. Gametes are represented inside a circle. And we know this is a heterozygous individual having capital T and small t. And following the law of segregation, they separate from each other so that each factor enters into each gamete. So, which is the factor which is going to enter into this gamete? It can be capital T. And in this circle or in this gamete, small t enters. Okay, again, the same thing happens over here. This is a heterozygous individual. When it was homozygous, all the gametes were similar. Whereas in a heterozygous individual, 50% of the gametes carry the capital T allele and 50% of the gametes carry small t allele. So, in order to understand this crossing or what are the random fertilization which is going to take place over here, let us represent a Punnett square. So, a Punnett square, I am going to draw a Punnett square. Okay, and the gametes. What are the gametes? It will be the pollen on one side and the eggs on the other side. And these pollen and gametes, that is the gametes, the pollen and the egg, they are represented on the top of the rows and at the side of the columns, which is represented as male and female gametes. Represented as male and female gametes. So now let us understand the probability. What all types of cross can take place over here in this hybridization experiment. So in the first possibility, the capital T and capital T can fuse with each other. That means each gamete over here, that if we take this as the pollen grain, the pollen grain carrying the capital T allele has 50% chance to go and pollinate the egg carrying capital T and 50% chance to pollinate the egg carrying the small t allele. And again, the pollen carrying the small t allele has 50% chance to pollinate the egg carrying the capital T allele and 50% chance to pollinate the egg carrying the small t allele. So now let us conduct the process. Over here we can get capital T, capital T. This cross we'll get capital T, small t. And this together we get again capital T, small t. And these two together we get small t, small t. So capital T is a homozygous condition. And what will be expressed over here? It will be surely what the phenotype will be tall. So let us express this or let us write this as tall. And again, this heterozygous condition, we know capital T is dominant over small t. And again we get the phenotype tall. This is also tall. Whereas small t homozygous. What is this phenotype? It is always dwarf. The F1 generation represented or expressed only one parental trait, that is the tallness. It did not express the dwarfness. One parental trait was completely missing. And the missing trait reappeared in the F2. And this is called as the F2 generation. This is called as the F2 generation. And the missing trait expressed or reappeared in the F2 generation. But what is the frequency? We can see tallness is 3/4. 3/4 of the F2 hybrids were tall. And only 1/4 of the F2 hybrid is dwarf. So the ratio of or the phenotypic ratio is 3 tall is to 1 dwarf. This is the phenotypic ratio. The phenotypic ratio is 3:1. 3 tall is to 1 dwarf. Now let us consider the genotype. What are the different possible genotypes that we have obtained in this F2 progeny? We can get 1 capital T. And how many capital T small t is a heterozygous condition? We get 2. 1 and 2. So 2 capital T small t and 1 small t small t. So this is the genotypic ratio. This is the genotypic ratio of a monohybrid cross. And the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. And from this cross, we can understand the law of dominance in which the F1 all are tall because in this dissimilar pair of alleles, one pair, that is one member of the pair, capital T, dominates the other member, that is small t. And the character which is expressed in the F1, that is tallness, is considered as a dominant character. And the other parental trait which is not expressed in the F1 but which was expressed in the F2 in a very small fraction, 1/4, is considered as a recessive character or recessive trait. The other law, that is law of segregation, in which a pair of alleles which is present in the parents segregate at the time of gamete formation so that only one member or one factor or one gene enters into one gamete. And a homozygous individual, all the gametes are similar, and in a heterozygous individual, 50% of the gametes carry one factor and 50% of the gametes carry the other factor.
Thank you.